It is my pleasure to introduce our next speaker, Dr. Igor Koturbash. Dr. Koturbash is an associate professor, and he is the chair of the Department of Environmental and Occupational Health and the director of the Regulatory Science Program at the University of Arkansas for Medical Sciences at Little Rock. He received his MD from the State Medical University in Ivano-Frankivsk in the Ukraine and his PhD in biomolecular sciences from the University of Lethbridge in Canada. The focus of his research is to understand mechanisms of tissue responses to radiation and how diet and dietary modification can modulate tissue response to radiation. He has published over 100 peer-reviewed articles in addition to several book chapters. He serves as an associate editor for the professional journal Radiation Research and is a recipient of the Michael Fry Award from the Radiation Research Society. It is my pleasure to present Dr. Koturbash.
Good afternoon, ladies and gentlemen. My name is Igor Koturbash, and I present University of Arkansas for Medical Sciences. And before I start, I would like to express my sincere gratitude to the organizing committee. It's been both pleasure and honor to present here. And today I'll be talking about the radiation epigenetics, understanding how we can use the knowledge of radiation and epigenetics in development of biomarkers of exposure, and how can we better understand the process of radiation carcinogenesis. Most of the slides will be devoted to the work I conducted probably in the last decade. And then without further ado, I will start my presentation by saying that ionizing radiation is a well-known DNA damaging agent. That is, that radiation causes all sorts of breaks to DNA, singles, double-strand breaks, there's a cross-linkage, and so on. Those effects are generally considered as direct effects of exposure to radiation. There are also indirect effects or actions that has been associated with formation of reactive oxygen species. However, radiation is also capable to cause non-direct DNA damaging effects, or so-called epigenetic or epigenetoxic effects. And the very first evidence of such effects were described by John Kalinich and his colleagues back in 1989, when they showed the changes in DNA methylation after exposure to gamma rays in different cultured cell lines. So before we go further into the DNA methylation and other epigenetic effects, let's get back to just understanding what epigenetics specifically is. Here's a little cartoon that would probably describe the epigenetic and mechanism of epigenetic regulations the best. So we have pretty much four major types of tissue. That is connective tissue, that is epithelial tissue, the muscle tissue, and the nervous tissue. So these four types of tissue comprise about 90 of different organs and systems that make us exist and function. In turn, these 90 organs and tissues have been built of about over 200 different types of cells. What's really interesting is that all these cells actually have the same genetic information encoded. So there is something about the nerve cell that it looks like a nerve cell and doesn't function as a nerve cell, whereas a hepatocyte that has the same genetic information encoded functions like the hepatocyte and look like hepatocyte. So there is something that regulates the expression of genetic information in the cell, tissue, and also sex-dependent manner. And this mechanism of regulation are known as epigenetic, or those that are not associated with a DNA sequence. So the most known and studied up to date effects, epigenetic mechanism is methylation of DNA, or the addition of a methyl group to the fifth position of carbon. During my early studies on epigenetics, and actually my PhD during my PhD studies, we were able to show the epigenetic effects of exposure to radiation in vivo. What was important to that is that not only we showed it happening in an animal model, but these changes can be stable by nature, persistent, and they are kind of more characteristic of radiation target tissue. For instance, if we look here at the thymus and the muscle, so thymus is clearly the target for radiation, whereas muscle is kind of more non-targeted tissue. So shortly after radiation, we see changes in levels of global DNA methylation in both tissues. It's a 6 hours time point. However, 1 month after radiation, there is no changes in DNA methylation in the non-target tissue in the muscle, but we could clearly still see persistence or presence of these stable changes in global DNA methylation in a thymus tissue. But this is when we're talking about global DNA methylation. Clearly, DNA methylation is not uniform, and it has certain patterns of distribution of methyl groups across the genome. So in order to better understand where does it happen, where it's coming from, is the next cartoon, which is kind of a little bit old already, but it's really nicely recapitulates the current understanding of patterns of distribution of methyl groups across the genome. So if we look at the panel A here, then one of the most common places where DNA methylation can occur is the promoter regions. Before the first exon. A lot of genes have so-called unmethylated CpG islands. CpG island is concerned a DNA sequence which has the CG frequency of about 55% on a duration of at least 200 base pairs. So, for example, here is an example of a gene that has quite high CpG size frequency, labeled in green. But there is no methyl groups there. So you can clearly see the transcription factor binding site is open to transcription factor to bind, and then RNA polymerase can initiate the transcription, meaning that this gene is being active. In disease, especially in cancer, loss of such genes, actually, there are methyl, those CpG islands, CpG sites within the island are being methylated. For example, we can see MBD or methyl binding domain present there, which attracts the DNA methyltransferases and methylates those CpG sites, which means that the initiation of transcription is impossible, and this gene is being silenced. Many of those genes have been silenced in cancer, and they're so-called the tumor suppressor genes, so those whose function in cancer is being impaired. Another option for methylation of DNA is presented in panel B, where methylation occurs about 2 kb in front of the promoter. So this is a so-called CpG island shore, when we have quite high density of CpG sites, about 2 kilobase ahead of the promoter of the gene. Apparently, the free methylation status of the CpG island predetermines the active status of a gene, while methylation of the CpG sites results in silencing of the gene, once again, very often that's been observed in cancer. Not only the promoter of the gene can be methylated, but also various areas within the gene body. And of this, specifically interested in the methylation within the intronic regions that are being usually heavily methylated, but can have alternative transcription start sites. So if these methyl groups are being lost and the alternative transcription start sites are being open for initiation, we often observe that alternative or spurious transcription of those genes. And very often it's very characteristic for oncogenes, those actually whose overexpression can result in cancer development and or progression. Last, but not clearly least, is the methylation of repetitive sequences. Why it's important? If you look at the gene body, at genes, they comprise 1 to maybe 2% of entire genomes. However, the repetitive sequences represent 50 and up to 70% of the entire genome, depending on the species. The repetitive sequences can be mobile, and they actually been mobile during all these years of evolution. And their unwanted transcription activity associated with further transposition, recombination, and genome instability is being maintained by methylation of DNA, which helps to silence them. Once again, in disease and very often in cancer, what we observe is a loss of methylation within repetitive sequences, which further results in a number of unwanted effects. Okay. One of the most interesting of those repetitive sequences that I will be talking mostly today is the long interspersed nucleotide element 1, or line 1, or L1 element, as very often being called. This is a repetitive element, which is the most ubiquitous among all the elements in any mammalian system, a mammalian genome, comprising about 17% of a human genome and about 22% of a mouse genome. So line 1 has been very heavily methylated, so which is that methylation mostly occurs within the 5' UTR of line 1. And this methylation actually prevents its unwanted transcription, as line 1 can make its own copies while it's being active and then introduce them somewhere else in the genome, which is called the insertional metagenesis. Exposure to various environmental stressors, and as we will see during today's presentation, exposure to radiation can often negatively affect the methylation of DNA of line 1 elements and result in their transcriptional reactivation and insertional metagenesis. So why all this is important? So once again, methylation of DNA is a key mechanism of expression, of regulation of expression of genetic information. That is, if we have altered DNA methylation, it's inevitably going to result in an altered gene expression. Methylation of DNA is also critical for silencing of repetitive elements. For example, altered methylation of line 1 can result in its reactivation, which further will result in insertional metagenesis, genome amplification, and subsequently development of genomic instability. Even without retrotransposition, DNA hypermethylation of repetitive elements can alter gene expression. For example, if we go back to the cartoon I showed on the previous slide with methylation within the gene body, very often line 1 can introduce its copy within the open reading frame within the gene body of an active gene, thus creating alternative transcription start sites and can result in spurious activation of the gene. And last but not least, global DNA hypermethylation, which is mostly associated with the loss of line 1 DNA methylation, it has been considered for about a couple of decades as a hallmark of cancer and one of the driving mechanisms of carcinogenesis. So let's move a little bit to experimental work and things that we did in the last probably 8 years on trying to better understand how radiation affects methylation of DNA, what's the involvement of line 1, and how can we use that knowledge in order to better understand the mechanisms of radiation carcinogenesis and development of reliable biomarkers of radiation exposure. So in this regard, of particular interest to us was the not terrestrial but space-like radiation. Terrestrial radiation represented as, for example, gamma ray or X-ray, they're mostly photons, and these are relatively low charged energy photons. When we talk about space-like radiation, they are mostly presented as the high charge, high energy particles. And for example, heavy iron ions is one of those great examples. So we had mice that had been exposed to a space-like radiation in this case, with the heavy iron ions. And so we looked at long-term effects. These mice were investigated 5 months after exposure. And so we used space environment relevant doses, or those that astronauts can experience during deep space exploration missions. So we see at a dose at 0.4 gray of heavy iron ions, we see significant increase in global patterns of DNA methylation. So what we think in this regard, if we see hypermethylation, so it's not probably associated with hypermethylated line 1 elements, but probably with the tumor suppressor genes that actually can acquire methyl groups and will become inactive during process carcinogenesis, thus contributing to that process. However, when we ran the tumor suppressor gene DNA methylation array, we didn't find any significant changes at 0.4 gray dose that was associated with global hypermethylation. However, when we looked at repetitive elements, starting from retrotransposons like line 1 and sign B1, the major and minor satellites, repetitive elements that are characteristic for paracentromeric regions of a chromosome, or some elder evolutionary transposons, what we saw that changes in DNA methylation were coming primarily from those repetitive elements. The assays in here were actually determining the extent of demethylated DNA. So the lower the bar, the higher extent of methylation of DNA. So in this case, we're observing hypermethylation of those repetitive elements at 5 months after exposure to space-like radiation. So next step, what we did, actually, we looked at real radiation target tissue, which is the hematopoietic system. And as we know, the most earlier radiation-induced cancers are those hematopoietic malignancies. So we use the same settings. Animals were exposed to low dose, relatively low dose of space-like radiation. Once again, in this case, was the heavy iron ions. But what we looked now, and not in the entire cell population, but we were able to sort the cells and have hematopoietic progenitor and stem cells, or HPCs on one hand, which are at the top of the pyramid of hematopoietic cell differentiation, and mononuclear cells, which actually are terminally differentiated cells. What we could clearly see, there is already difference in DNA methylation between these two types of cells, which was mostly driven probably by differential expression of DNA methyltransferases, or those genes that are responsible for copying methylation patterns to the daughter strand, or methylation of newly created methylation sites. So when we looked at global methylation patterns in hematopoietic stem cell, hematopoietic and progenitor and stem cells versus mononuclear cells, we could clearly see lack of changes in mononuclear cells, but increase in methylation, in DNA methylation of hematopoietic progenitor and stem cells. However, when we looked at the methylation of repetitive elements, we could clearly see that the extent of demethylated DNA in hematopoietic progenitor stem cells was also increasing. So that was really a very puzzling effect to us, seeing increase in global DNA methylation, however, decrease in methylation of repetitive elements. So in order to understand this discrepancy and explain it, so we went back to understanding the architecture of these line 1 elements. So every line 1 element actually has two open reading frames, ORF1 and ORF2. It has the 3' UTR that has been followed by poly A tail. And the promoter region is built over 5' UTR monomers and can often be over 1 kb long, apparently sometimes even to 2 kilobase long. What's really very interesting is you can see that the bodies, both open reading frames of line 1 elements, have relatively low number of CPG sites, but most of them actually are being gathered at the 5' UTR areas. So unfortunately, the existing methylation assays for line 1 elements at that time were actually based on open reading frames and certainly could not depict what's happening on a global level on 5' UTRs, where the most of the CPG sites in line 1 are being located. Further, line 1 elements are not really uniform. They were evolving parallel with evolution, where one family of line 1 elements has been active for some time, then becoming extinct and replaced by another family that would once again will be existing for a certain time and being replaced by the next family. And in total, for example, in the mice, we observe almost 3 dozen of those families, and about the same number of those families actually exist in humans. And what's interesting that the difference between those families was observed not within the open reading frames, as these sequences were relatively conserved from the very oldest to the very youngest repeat line 1 element. But the differences were coming specifically from the 5' UTRs. So when we started looking at methylation of line 1 elements, depending on their evolutionary age, for example, from one of the most oldest L1LX3 line 1 element, and compared it with the younger elements like LMD1A1, we could clearly see a very significant difference in their methylation patterns. So the older the element, the lower DNA methylation of it, and the younger the element, the higher methylation of it. Why does it happen? There are probably several explanations, and probably the easiest to say that, well, as these elements are being extinct, they probably their methylation of their DNA is probably rudimentary, and you don't need to silence something that actually is not being active anymore. But in my opinion, the most plausible explanation is that the cytosine, during many years, millions of years of evolution, the process of spontaneous mutation actually occurs on a regular basis. And cytosine is the most mutatable base in this regard. For example, if you have CG sequence, it can be methylated. If that cytosine becomes adenine and now it's AG, it cannot be methylated anymore. So as you lose those cytosines and being replaced with, for example, adenine and so on, it cannot be methylated, and thus we observe this progressive loss of DNA methylation. So having explored this, we went back to our space radiation models, and we had mice that were exposed to low, once again, the space environment relevant doses of either protons or heavy iron ions, or combined radiation with protons and heavy iron ions. And what was interesting that on a global level, for example, we did not see any changes in global DNA methylation there whatsoever. When we applied Pyrus sequencing assay to look at specific methylation of line 1 elements, we saw a small but significant decrease in methylation of line 1 in groups where we had protons, so either proton alone or proton plus heavy iron ions radiation combined. But once again, this assay was based on ORF1, right? The area which is not really being heavily methylated. When we applied our array looking specifically at methylation of different line 1 elements depending on their evolutionary age, from a 10 million old element L1LX3 to the youngest one, we could clearly see that there were not uniform patterns of DNA methylation, but we could clearly see that the elements, for example, the oldest one, were not really significantly, did not experience significant changes in methylation, as most probably they don't really have too many CG sequences to be methylated or to lose methylation to start with. The younger elements are being heavily methylated anyways to prevent their unwanted transcription. So the changes what we observe are primarily coming from the element that is being pretty much somewhere in the middle of its evolutionary age, that had probably a lot of CG sites still available that could acquire this methyl group. So for example, this guy LM1MDA2. So having seen that, what we were very interested to see now, applying all this knowledge, looking at terrestrial radiation, the radiation that is more applicable to most of the humans, and trying to better understand the methylation patterns, especially in regards to line 1, after exposure to low doses of radiation using 2 different mouse models in a hematopoietic or radiation-sensitive tissue. We use the C57 Black 6 mice. These mice are being considered relatively radio-resistant. They not readily develop radiation-induced tumors. And CBA mice. These mice are being considered as relatively radiosensitive, as they readily develop radiation-induced hematopoietic malignancies. So we exposed these mice to either 0.1 gray, which is really low dose radiation, or 1 gray dose of radiation, which is kind of the lowest dose at which the CBA mice are known to develop radiation-induced hematopoietic malignancies. And then we looked at hematopoietic stem cells, which are on the very, very top of the hierarchy of differentiation in the hematopoietic system. This is the mostly undifferentiated cells that actually give rise to all the next populations of hematopoietic cells. We have one step down from hematopoietic stem cells, the hematopoietic progenitor cells, and we have terminally differentiated mononuclear cells, those that at the last stage of differentiation, those are functionally specialized cells that actually are present in our blood and whose life cycle is about 100 to 150 days. So what was really very interesting to see, in a radio-resistant strain, we did not observe any significant changes in methylation of line 1 elements, independently whether we're looking at hematopoietic stem cells, progenitor cells, or mononuclear cells. The difference where we see is coming from a radiosensitive CBA mice. For example, in the hematopoietic stem cells, we could clearly see a significant loss of DNA methylation in line 1 elements independently of their age. We could clearly see dose dependent, where the smaller changes associated with exposure to 0.1 gray were followed by much more deeper hypermethylation observed with exposure to 1 gray. Those changes were way less pronounced in more differentiated progenitor cells, and actually were pretty much absent in mononuclear cells, with only some hypermethylation patterns observed after a high dose. But once again, these changes do not have a long-term consequences, as these cells will be fully replaced within 100 to 150 days, while the hematopoietic stem cells are becoming epigenetically reprogrammed, and they will give rise to epigenetically reprogrammed and altered hematopoietic stem cells, hematopoietic cells throughout the life of this individual. So altogether, these changes suggested that line 1 methylation is a very interesting tool to better understand the mechanisms of radiation-induced carcinogenesis, but they also can serve as the biomarker of previous exposure to ionizing radiation. So all this evidence, what we had, was actually coming from animal studies, and very often what we see in mice or rats or other experimental systems is not applicable to humans. So certainly we need to find whether what we see in an animal will be applicable and translated, validated in humans. So in this regards, I will share some details on our most recent study where we looked at molecular biomarkers of radiation exposure in Marshallese. Most of you probably aware that back in middle of the 20th century, around 1950s, a number of nuclear weapon testings were performed at Marshallese Island back in the Pacific Ocean. And you can see there are 4 atolls where the most of tests were performed. However, However, the radiation plume was further moved not beyond these atolls, and much larger fraction of atolls were exposed, resulting in thousands of Marshallese being exposed to relatively high doses of ionizing radiation and for a prolonged time. So what's known about the Marshallese is that there is increased cancer incidence and metabolic syndrome in residents of Republic of Marshall Islands. We also know that a lot of radiation-induced cancers are still expected to emerge because they have a very long latency, as radiation-induced cancers sometimes takes several decades to emerge. And also, as we know from studies from Dr. Dubrova, that a lot of radiation-induced effects can be transmitted to the next generation. So there is significant, there is quite compelling data on cancer incidence in, for example, thyroid cancer in Marshallese. There is also studies indicating hematopoietic malignancies as well as colon cancers that are associated with exposure to radiation. So a large group of Marshallese actually relocated to Northwest Arkansas back in 1970s and throughout early 90s, and now we're experiencing a new wave of immigration of Marshallese to Northwest Arkansas. And at this moment, about 10% of the entire population of Republic of Marshall Islands actually reside in Northwest Arkansas. There's lots of studies that indicate health disparities in Northwest Arkansas Marshallese. For example, they have very high level of diabetes and cardiometabolic syndrome. And these health disparities are being largely attributed to socioeconomic and genetic factors. However, it remains unclear what the specific role of previous radiation exposure in the etiology of this disease in this group of people. So what we hypothesize that this increased cancer rates in the development and the development of metabolic syndrome in Marshallese actually are related to those exposures that Marshallese experienced as acute exposure from radioactive fallout, premature resettlement to some of those atolls when people were told, well, it's safe to go back, but apparently it was not, and also associated with the long-term consumption of contaminated locally grown food, as these people living in such remote destinations were mostly dependent on local drinking water and locally grown foods. So what we did, actually, we had a residential history and geography of the study participants. And what we did, we divided them into two categories, where we had the North group, to which study participants belong, that spent at least 6 months on the northern atolls, north from Kwajalein and north from Kwajalein, and that they spent this time before 1980s. Why 1980 is that we took into consideration, first of all, the half-life of cesium, which is about 30 years, right? So we have to take into account the radioactive decay of cesium, which was the most radioactive contaminant from the fallout. There are also major cleanup efforts at the end of the 70s that helped to reduce the number of environmentally present cesium. And also because in the 80s, the potassium soil treatments were introduced. What potassium does, it's a natural competitor for cesium, and introducing potassium into the soil will significantly decrease the root uptake of cesium from the soil by plants, and thus will significantly decrease consumption of radioactive cesium-contaminated foods. So the people who actually were before 1980s and spent more than 6 months on the northern islands, those that actually were mostly contaminated belong to our North group. Whereas the South group were citizens who represented those who lived most of their lives in the South, and they did not spend more than 6 months on the North atolls throughout their entire life. So we started from studies of chromosomal aberrations, as this is clearly the golden standard at this very moment. And here is just a representative figure to show you how the normal on the left side, and whereas an anomalous on the chromosome 6 can look. So when we look specifically quantitatively at chromosome rates, so we could chromosomal aberration rates, we could clearly see that participants that spent 6 or more months in the northern atolls actually had significantly higher aberration rates compared to those participants that actually spent on the south. And this difference was primarily driven by women study participants. When we actually plotted the frequency of aberrations per 10-year time periods, we could clearly see that every 10 years spent in the northern atolls before 1980s corresponded to a significant 0.31 increase in aberration rates. So telling us the longer you lived in the northern atolls, the higher probability of developing chromosomal aberrations. So what we looked then at the methylation of those line 1 elements and other repetitive elements, and we tried to correlate these changes in DNA methylation with chromosomal aberrations. So similarly to mice, we could clearly see that there were changes in DNA methylation were not really uniform, with some repetitive elements acquiring methyl groups and some actually having a decreased methylation. But what was really very important to identify certain repetitive elements, and once again you can see that all of them were actually the various line 1 repetitive elements, that the methylation status of these repetitive elements was inversely correlated with number of chromosomal aberrations. That is, the lower methylation of DNA in that locus, the higher number of chromosomal aberrations were observed in these study participants. So in conclusion, I would say that, first of all, ionizing radiation, besides being a potent genotoxic stressor that is affecting the sequence of DNA, can also affect the cellular epigenome. Further accumulating evidence suggests that epigenetic alterations actually are being involved and actually the important mechanisms of cancer development and progression. We know that at least some of these epigenetic alterations are stable by nature. They can be detected long-term after exposure to radiation. We know about the significant correlation between the chromosomal aberrations and DNA methylation of specific line 1 elements. And that all tell us that besides being an excellent tool for the studies of radiation epigenetics, line 1 holds a very good promise for the development of biomarkers of previous exposure, since radiation leads to long-term persistent alterations in the pool of target cells. At the end, I would like to certainly acknowledge all the people who are involved in the studies, people from my laboratory, some who were already postdocs and some who came as PhD students and actually got their doctoral degree doing this work. I would like to thank my colleagues at UAMS, and especially Dr. Martin Hauer Jensen. And I would like to acknowledge also our collaborators on works on space radiation, Dr. Raber from Oregon and Dr. Nelson from Loma Linda. And certainly all this research would not happen without precious funding from NIH, NASA, and Arkansas Biosciences Institute. So once again, thank you so much, Time, for giving the opportunity to talk today, and I will be happy to accept any questions. Thank you.